The second major energy system is the anaerobic glycolysis energy system. CHO stands for carbohydrate. Remember, among the three macronutrients, only carbohydrate can involve and play a role in glycolysis system. So in the glycolysis system, we don't see fats like triglycerides or fatty acids, and also we don't see proteins, only carbohydrates. And in this case, it's glucose. So in this reaction in the anaerobic glycolysis system, again, it's anaerobic, which means without oxygen. We get lactic acid, and this energy system produces two ATPs. And from the lactic acid, we get lactate and one hydrogen ion. So this is the overall view of the glycolysis system. But we will talk about that in detail. So here in this figure on the right side of the slide, as you can see, we have glucose, which is the starter of the anaerobic glycolysis system. And from glucose, we have like instead of creatine phosphate, which was a one chemical step, we have a few steps here for the glycolysis system. In other words, creatine phosphate was very fast and very short in terms of the duration, but this one it's not very short, it's just short. Like it's shorter than oxidative phosphorylation, but it is like it takes longer for this energy system than creatine phosphate. Because instead of just one chemical step, we have a few steps here. I don't want you to remember and to memorize the name of the enzymes here. I will let you know what enzymes are very important so you can keep those in mind. But just this is an overview, and as you can see, at the end we get two ATPs because we are using two ATPs at the top of the glycolysis system. And at the end, we produce four. So the net ATP production is two molecules of ATP through glycolysis system. And at the end, we get lactate or lactic acid. So this is anaerobic glycolysis. It's also called the lactic acid system. So if you see this term in like research articles, or other textbooks, lactic acid system, that's another name for anaerobic glycolysis energy system. It's a series of chemical reactions that break down glucose to rephosphorylate ATP to ATP. We don't need oxygen, that's why it's anaerobic. And the final product is lactic acid, which is an organic compound, just like creatine. It is an organic compound because it contains carbon atoms in its structure, chemical structure. Glycolysis system contains and consists of 18 chemical reactions or chemical steps and 12 chemical compounds, and six reactions are repeated. So we see 12 different chemical compounds in this energy system, and six of the 18 chemical reactions are repeated because, let me explain it here. Glucose is a six carbon molecule, and here we have pyruvate. Pyruvate is a three carbon molecule. So in this case, glucose can be divided into two 3-carbon molecules. That's why the some of the chemical steps are repeated. So, but overall, we have 18 chemical reactions from breaking down of one glucose. And the lactic acid can be converted to a lactate and a hydrogen ion. And we have glycogenolysis, which is the breakdown of liver glycogen to glucose. So this glucose, our starter of the glycolysis system, the glucose can come from the breakdown of liver glycogen, which is called glycogenolysis. This lysis part of the glycogenolysis means breaking down. So, for example, we have glycolysis system, which means breaking down of glucose, and glycogenolysis means breaking down of glycogen. So, whenever you see lysis at the end of a word or a term, it means breaking down. So, and each chemical step is catalyzed by an enzyme. Recall from the creatine phosphate. That was just one chemical step, but we had one enzyme, which was kinase, creatine kinase, or CK. So here in the glycolysis system, each chemical step is also catalyzed by an enzyme. And in every chemical reaction which requires enzyme, there is a rate limiting enzyme, which is an enzyme that controls the speed of all the reactions. And with glycolysis energy system, that's phosphofructokinase, PFK. PFK is the rate limiting enzyme for the glycolysis system. Glycolysis produces 4 ATP, but 2 ATP are used in the process. So again, you can see from the red, um, like ATP, they are highlighted in red. Those are the ATPs that are used by the glycolysis system at the top of the chemical reaction on the right side of the slide. So glycolysis uses 2 ATPs, but at the end it produces 4 ATPs. So the final or net ATP production from anaerobic glycolysis is 2 ATP molecules. Glycogenolysis is more energetically efficient. Why? Why do you think the glycogenolysis is more energetically efficient? Like from glycogenolysis, why is it energetically efficient? Just one molecule of glucose produces 2 ATP molecules. But if we break down glycogen, you see here again on the right side of the slide, when we have glycogen, we skip the first step, which requires ATP. So if we break down glycogen through glycolysis system, we only use one ATP instead of two ATPs. So the final ATP production from glycogenolysis is 3 ATP molecules instead of 2 ATP molecules. So just keep that in mind. So from glycogenolysis, we get 3 ATP molecules because we skip the first chemical step from glucose to glucose 6 phosphate. If we break down glycogen, we skip the first chemical step, so we produce more ATP molecules. And as I said, like for creatine phosphate, each energy system has some advantages and disadvantages. Here is the major disadvantages of glycolysis system. After a while, it increases the acidity or decline in pH, which is also called metabolic acidosis, during moderately high to high intensity exercise. Why it increases acidity? Because it produces lactic acid, and that is an acid, and that acid leads to decline in pH or causing acidity or metabolic acidosis. So that's one major disadvantage. And this metabolic acidosis can also cause the decrease in activity of key metabolic enzymes. Remember from your physiology or other like basic nutrition classes, enzymes are proteins. They have a protein structure and their activity depends on several factors, one of which was temperature, another one is acidity or like pH. So enzymes has they have an optimal activity during some specific range of pH. But in the case of metabolic acidosis, we have a decrease in pH or acidity. So this can cause some problems for the activity of key enzymes inside the cells. So that's one major disadvantage of glycolysis system. And this decrease in activity of metabolic enzymes can cause muscle fatigue. From the creatine phosphate, we also get muscle fatigue when we had depletion of creatine phosphate levels. But regarding the glycolysis system, because of the production of lactic acid and accumulation of the lactic acid can cause acidity, and from there we can get muscle fatigue. The duration of this system is 1 to 2 minutes. Um, it is longer than creatine phosphate. It was just 10 to 15 seconds for the creatine phosphate, but this is for 1 to 2 minutes. And to fight against metabolic acidosis, we can we can take some dietary supplements like bicarbonate as a dietary supplement. And this supplement enhances the body's buffering system, which means it fights against the acidity because it has an alkaline characteristic and feature. It can fight against acidity or lactic acid. Some kind of it's like eliminate the lactic acid. So this is one strategy to fight against metabolic acidosis. And this bicarbonate supplement can cause some problems like gastrointestinal problems, cramping, bloating, vomiting, and diarrhea. For the anaerobic glycolysis system, we have several different types of exercises that predominantly uses glycolysis system. For example, 400-meter sprint in track. We have intermittent sprints, soccer, basketball, or like 10 to 15 repetitions of weightlifting, 50 to 100-meter swimming intervals, 300-yard shuttle run, and wrestling. These are the type of exercises that predominantly use anaerobic glycolysis system. Remember, this is just predominantly using one energy system. It doesn't mean that these exercises do not use other energy systems. They use other energy systems too, but they use anaerobic glycolysis system as their major energy system to replenish ATP. And here is the summary of the anaerobic glycolysis system. So we have 18 chemical steps or reactions. Six are repeated, 12 chemical compounds, and 11 enzymes are involved. The rate limiting enzyme is phosphofructokinase or PFK. It's fast, but not as fast as the creatine phosphate system. It's again anaerobic, just like creatine phosphate. Duration is 1 to 2 minutes, and the fatigue is associated with decreased pH. And it's a predominant energy system in high intensity exercise, for example, sustained repeated sprints. Let's answer this question. Glycolysis produces how many ATP molecules, but these ATPs are used in the process, so the final net ATP production from anaerobic glycolysis is 4 to 2. So glycolysis produces 4 ATP, but 2 ATP are used in the process, so the final ATP production from anaerobic glycolysis is 2 ATP molecules. If it was glycogenolysis, it was produces 3 ATP, but 1 ATP are used in the process. So the final ATP production from glycogenolysis is 3 ATP molecules. Here is the fate of the lactate. So from the glycolysis, we get pyruvate. From one molecule of glucose, we get two molecules of pyruvates. Again, as I said, glucose is a 6 carbon molecule, but pyruvate is a 3 carbon molecule. So from one molecule of glucose, we get two, 3 carbon molecules of pyruvate. And this pyruvate can be converted to lactate. And also lactate can be converted to pyruvate. So this is a two-way reaction. From pyruvate to lactate is a two-way reaction. It can happen from pyruvate to lactate and from lactate to pyruvate. But this lactate also can uptake from liver, heart muscle, slow twitch muscle fibers, and kidney, and use the lactate for aerobic phosphorylation. So the lactate molecule is used by cells in the body as a fuel source, and the pyruvate can be metabolized either anaerobically or aerobically. So if the pyruvate be converted to lactate, it's called anaerobic. But if pyruvate enters to the Krebs cycle, which we'll talk about that later, it's called aerobic. So this is the final fate of lactate. So lactate can also be used as a fuel source, as an energy source by different tissues like liver, heart muscle, kidney, and slow twitch muscle fibers. So anaerobic glycolysis relies upon the aerobic energy system to metabolize lactate. Just like the creatine phosphate, remember we talked about the creatine uh, uh, phosphate shuttle, which was depending on the aerobic energy system. So in the anaerobic glycolysis, In order to metabolize lactate, we use aerobic energy system. So from here we have, this is our muscle fiber, for instance. We use glucose or glycogen um, in the glycolysis energy system, and we produce lactate from that system, and this lactate enters the blood, and from blood can be uptaken by heart, liver, kidney, and slow twitch muscle fibers. And these tissues can use lactate as a fuel source. And lactate can be converted to glucose. It's called gluconeogenesis in the liver. So lactate can be also used for making glucose in the liver. So this is very important, just in the liver. Gluconeogenesis is a process of uh, building and synthesizing glucose from other compounds, and lactate can also be used to create and to synthesize glucose in the liver. And the process is referred to as the Cori cycle. So the Cori cycle is there are some like a couple of chemical steps and reactions, and that's called the Cori cycle, in which lactate be converted to glucose in the liver. And let's talk about the third and our last major energy system. It is the oxidative phosphorylation system. For the so far we talked about two energy systems. The first one was creatine phosphate, in which we use creatine. The second one was glycolysis system, anaerobic glycolysis system, and the starter molecule was glucose or glycogen, which are carbohydrates. For the oxidative phosphorylation, we can use all three macronutrients. We can use carbs, we can use fats, and proteins. And these macronutrients can be used in a cycle called Krebs cycle, an electron transport chain, which then we produce ATP molecules. We replenish the ATP molecules through the oxidative phosphorylation. So oxidative phosphorylation is an aerobic energy system, which means it depends on oxygen. And it's a process by which carbs, fat, and proteins can be metabolized to release the energy necessary to phosphorylate ATP to ATP. And the duration is 2 or 3 minutes to many hours. So unlike the creatine phosphate and glycolysis, which were too short or short, this is long. It takes time for oxidative phosphorylation. And one reason why it takes time, we have so many chemical steps and reactions in the oxidative phosphorylation. Remember, creatine phosphate had only one chemical step, and glycolysis was 18 chemical step. But the Krebs cycle and the oxidative phosphorylation is more than 30 chemical steps, so it takes time. But we can use the oxidative phosphorylation for long um, and low intensity exercises. So the duration is 2 or 3 minutes to many hours. And here on the right side, we have glycolysis energy system at the top, which from one molecule of glucose we get pyruvate, and this pyruvate can be used in a oxidative phosphorylation system. So pyruvate, like the chemical reactions we have above pyruvate is anaerobic, but the bottom of the chemical reactions at the bottom of the pyruvate, it's aerobic. It depends on oxygen. So this is one fate of pyruvate, which we produce through glycolysis system. One fate of pyruvate is to be used in the Krebs cycle and in oxidative phosphorylation energy system. And remember from the beginning of the chapter, we overviewed the three energy system. We said creatine phosphate and glycolysis system takes place in cytoplasm of cells. Oxidative phosphorylation takes place in mitochondria. And we have three phases of oxidative phosphorylation. The first phase is preparation. The preparation of macronutrients like glycogen or starch or glucose or whatever, or preparation of fatty acids or triglyceride, preparation of proteins, amino acids. That's the first phase of oxidative phosphorylation. The second phase is called Krebs cycle. This cycle on the right side, you see, this is a cycle called Krebs cycle. Also, it's called citric acid cycle, uh, or TCA cycle, 3 carboxylic acid cycle, TCA. So if you see these names and terms, these are all referred to oxidative phosphorylation, Krebs cycle, citric acid cycle, and TCA cycle. The reason why we also call it citric acid cycle is the second compound that can be produced from oxidative phosphorylation. So that's why citric acid cycle. So citric acid is the name of the compound that is produced by the Krebs cycle. And the third phase is the electron transport chain, which takes place again inside the mitochondria. And we talked about that. We will talk about it later. So there are three steps or three main phases for oxidative phosphorylation. If the starting point is glucose, as we see from the figure on the right side, the entire oxidative phosphorylation from the preparation, Krebs cycle, and electron transport chain, we get 124 steps. So as you can see, this is very long compared to creatine phosphate and glycolysis energy system. So if we start from glucose, we have 124 steps, including the preparation, Krebs cycle, and electron transport chain. We produces and we uses overall in total 30 chemical compounds, and this entire energy system requires 27 enzymes. And the Krebs cycle, very limited ATP produced through the Krebs cycle. And the primary function of the Krebs cycle is to oxidize, which means removing electrons from the compounds produced from the breakdown of carbs, fats, and proteins. So from the Krebs cycle, which is our second phase of oxidative phosphorylation, very limited ATP are produced, and the main function of the Krebs cycle is to oxidize our macronutrients. On the other hand, we have electron transport chain, and we get many ATP molecules from electron transport chain. And why we call this electron transport chain? Because it's passing electrons through a series of chemical reactions. And the energy released from these chemical reactions used to rephosphorylate ADP to ATP. And the final electron acceptor is oxygen. That's why this oxidative phosphorylation depends on oxygen. And that's why we call it aerobic energy system, because the final electron acceptor is oxygen. We get three end products from oxidative phosphorylation: ATP, water, and carbon dioxide or CO2. That's why when we exhale or when we breathe out, we breathe out CO2 or carbon dioxide. This carbon dioxide is produced from oxidative phosphorylation. We also get water molecules from oxidative phosphorylation and ATP. These are the three end products of oxidative phosphorylation. So in this figure, this is a summary of the entire oxidative phosphorylation. So after glycolysis, again, glycolysis means breaking down of glucose. So we break down glucose and we convert it to pyruvate, and two molecules of pyruvates are produced from glycolysis. And then these pyruvates, glycolysis takes place in cytoplasm. And the pyruvates we get from glycolysis are also in cytoplasm. So these pyruvates are shuttled into the mitochondria, which we get to our second phase of oxidative phosphorylation, which is the Krebs cycle. A high energy phosphate compound is produced by the Krebs cycle and it's called GTP or guanosine triphosphate, GTP. And this is another high energy phosphate compound. So far we have learned about three high energy phosphate compound. One is creatine phosphate, two is ATP, and this is the third one that we have learned so far. GTP is another high energy phosphate compound. So this GTP is produced from Krebs cycle. And in total, from one molecule of glucose, we get 30 ATP from just one molecule of glucose. This is not a biochemistry class, this is not a physiology class, so I don't want you to remember the name of the enzymes or remember the chemical reactions and so on and so forth. Just to remember and learn the basics of the oxidative phosphorylation and other energy systems will suffice for this course. Like uh, where do the energy systems take place, or how many ATPs we can get from oxidative phosphorylation. So these all matter for this class. Like whatever we cover on the slides, that's all you need to know and you need to learn. Um, you don't have to be worried about the name of other compounds or enzymes. So let's define some terms. When we say oxidation, it's the process of giving up electrons. That's called oxidation. And remember, the primary function of the Krebs cycle was oxidize or oxidation. And when we say oxidized, it's the compound that has electrons removed or has been oxidized. So when we say oxidation, it's the name of the process of giving up electrons. And when we say oxidized, it's the compound that has been oxidized, or the oxidation happens or occurs to that compound. And we have reduction on the other hand. Again, this is also the name of the process, but this is the process of accepting electrons, not giving up electrons. And reduced is the compounds that receive the electrons have been reduced. We have also free radicals. The free radicals are a highly reactive molecule with an unpaired electron, also known as reactive oxygen species or ROS. You may see this in articles, in research papers, ROC, or oxidative stress. This is free radical. So any compound or molecule that has an unpaired electron is a free radical, and they are dangerous, they are harmful, they can cause many different problems, from oxidative stress to inflammation to cancer, so on and so forth. So these are the main terms that it's better to know these: oxidation, oxidized, reduction, and reduced. And oxidation reduction reactions are aspect of chemical reactions. So with the Krebs cycle, which is the second phase of oxidative phosphorylation, remember we said the starter molecule of glycolysis is glucose. The starter molecule of the Krebs cycle is a compound called acetyl-CoA. The acetyl-CoA contains two carbon. Glucose is a six carbon molecule, pyruvate is a three carbon molecule, and the acetyl-CoA is a two carbon molecule. And it begins the Krebs cycle. It's our starter. The Krebs cycle itself is a series of 10 chemical reactions, and we have six oxidation reduction reactions beginning with pyruvate. The most common electron accepting compound in the Krebs cycle is called NAD, nicotine amide adenine dinucleotide, or vitamin niacin. Vitamin niacin is another name for NAD. Like NAD is a derivative of vitamin niacin. Vitamin niacin is vitamin B3. And also we have FAD, flavin adenine dinucleotide, which is a derivative of vitamin riboflavin or vitamin B2. That's why the B vitamins are important in metabolism of carbohydrates and other macronutrients. And in vitamin B deficiencies, people get fatigued, they lose energy, they feel dizziness, that's one of the reasons. So these two vitamins, B3 and B2, are the coenzymes or are the precursor of electron accepting compounds in the Krebs cycle. So remember these, we have NAD and FAD in the Krebs cycle, and they play a role in four of the oxidation reduction reactions. So we have six oxidation reduction reactions in the Krebs cycle. Four of them are depending on NAD and FAD. And these molecules shuttle the electrons to the electron transport chain. So NAD and FAD are getting electrons from compounds in the Krebs cycle, and they shuttle the electrons to the electron transport chain, And from the electron transport chain, which passes the electrons through a series of chemical reactions, we get ATP. The rate limiting enzyme in the Krebs cycle is IDH, isocitrate dehydrogenase, IDH. That's the rate limiting enzyme. You know, from the glycolysis energy system, that was PFK, but here it's IDH. That's the rate limiting enzyme. The electron transport chain is our third phase of the oxidative phosphorylation, and it takes place within the inner membrane of the mitochondria. The oxygen is the final electron acceptor. It picks up the electrons in the form of hydrogen and forming water. The rate limiting enzyme in the electron transport chain is cytochrome oxidase, COX, cytochrome oxidase. That's the rate limiting enzyme. So in a skeletal muscle, FAD is responsible to shuttle the electrons into the mitochondria and not NAD. And the ATP yield as 2.5 from NAD and 1.5 from FAD. And this is the electron transport chain. As you can see, this is the membrane of the inner membrane of the mitochondria. And these are different molecules, protein molecules. This is called electron transport chain because it's like a chain that transport electron through these molecules. And when we transport and when we pass electrons through these molecules, we get energy. And from that energy, we can produce ATP. Here at the end, we get ATP. So one molecule of NAD can produce 2.5 ATP molecules, and FAD can produce 1.5 molecules of ATP. And this table summarizes the amount of ATP molecules we get from one molecule of glucose. You don't have to remember these numbers, but these are important, just to know the main differences. That's all. All right, let's talk about the physical activities and oxidative phosphorylation. So at rest, when we are resting, we predominantly use oxidative phosphorylation. In low to moderate intensity exercise, we use oxidative phosphorylation. Recall from two energy systems that we talked about, the creatine phosphate and the glycolysis energy system. We use those energy systems in high intensity exercises. But in low to moderate intensity exercise, we predominantly use oxidative phosphorylation. And these are the examples of these types of exercises, like walking, jogging and distance running, cycling, specifically for longer distances, swimming for longer distances, and dance aerobics. And here's the summary of the oxidative phosphorylation. We have 124 chemical steps or reactions, 30 compounds or 27 enzymes. The rate limiting enzyme The PFK is for the glycolysis part of the oxidative phosphorylation. And we have IDH and COX, these are the rate limiting enzymes. We get 30 ATP from glucose and 31 ATP from glycogen in a skeletal muscle. It's a slow energy system. It potentially limitless duration. As I said, when we rest, we use oxidative phosphorylation. So it has a limitless duration. It's aerobic, it depends on oxygen because the oxygen was the final acceptor of electron. And the fatigue is associated with fuel depletion. For example, muscle glycogen depletion. That causes fatigue. And it's the predominant energy system in endurance exercise because the endurance exercise are like they happen in like very long, like long distance running, long distance cycling, long distance swimming, and low to moderate intensity exercises. Let's match these with this. First one. It is shuttled to mitochondria after glycolysis. It's pyruvate, yes. Pyruvate gets converted to this intermediate compound. It's acetyl-CoA. Remember, pyruvate was the one of the last compound in glycolysis. And when we start the oxidative phosphorylation, we have to add pyruvate to be converted to acetyl-CoA, which was the starter of the Krebs cycle. So pyruvate gets converted to acetyl-CoA as an intermediate compound. The final electron acceptor is oxygen. That's why we call the oxidative phosphorylation aerobic. This is formed during the Krebs cycle. It's citric acid. That's why the Krebs cycle is also called citric acid cycle. So far we talked about three main energy systems. Creatine phosphate energy system. We talked about um, glycolysis and oxidative phosphorylation energy system. Next, we are going to talk about the fuel utilization.